Good evening, good afternoon, or morning from wherever you're joining us from today for our live webinar on the high-quality laboratory testing to transform the response to the global pandemic. My name is Alexandra, and I'll be moderating today's session. We are excited to have you all here today with us and hope you are staying safe wherever you are located. Today we have Jellica Wold, who is a Senior Manager of Immunoassay and Chemistry for Strategic Marketing for Beckman Coulter's global team. Jellica is a healthcare solutions marketer with over 10 years in product development and management. She has experience for medical implantable devices and diagnostics. She collaborates with medical professionals, defining any of their unmet needs. She also works on cross-functional teams to provide better solutions to healthcare problems. She has developed and implemented therapy awareness and educational programs around the globe. Jellica has a Master of Business Administration and Masters of International Management from the University of St. Thomas in St. Paul, Minnesota. She also has a Masters of Business Administration on Consumer Psychology and Behavioral Marketing and a Bachelor of Science in Chemistry and Biology from the University of Paderborn in Germany. Some of her many accomplishments include holding multiple patents for surgical tools, global commercialization manager for Medtronic in the past, and a senior global product manager for upstream for apps. So in today's objectives, we will be covering clinical utility of serology versus antigen testing, barriers to mass testing, low-touch testing, confidence in results, and the impact of mutations to current testing options. We ask you to send through your questions through the webinar chat box, and we'll address them at the end during our question and answer session. Now, I will thank you for your attention, and I'll turn the control over to Jellica. Thank you and have a great session.
Good morning, good afternoon, and good evening. Thank you very much for dialing in and joining us today. I'm honored to be here with you and to give you a little bit of information about the COVID menu that matters that Beckman Coulter is trying to offer to all of our customers across the globe. As Alexandra mentioned, we have a variety of topics that are going to be addressing high quality laboratory testing that we hope will assist you in curbing the spread of the global pandemic and transform the global pandemic response. As a disclaimer, let me just say that the information about medical practice recommendations from CDC and/or the WHO that is included in my slides is not to be interpreted as authorized use of the IVD assays. Intended use of the assays from Beckman Coulter is governed by the FDA and other regulatory bodies. Beckman Coulter assays are limited to the authorized intended use found in the product labeling, so we will have to stay on label in answering all of the questions that you may have as we conclude the presentation. The agenda, Alex already introduced us to, so I won't go over this in detail, but dive right into the presentation itself. COVID-19 has stopped the things we knew how to do prior to 2019. As of yesterday, there have been 112 million infections globally. 81% of infected people are asymptomatic, so they don't even really know they could be spreading the disease. Some of the 81% have mild to moderate symptoms. 14% of infected people show severe symptoms, however, and need treatment by a healthcare professional and potentially hospitalization. Out of the 14% who show severe symptoms, 5% of patients may end up in the ICU with dire consequences. As we have all learned over the past year, SARS-CoV-2 is a very aggressive virus that can be transmitted through aerosols. It targets the ACE2 receptors. SARS-CoV-2 shares about 79% of the sequence identity with the previously circulating virus of SARS-CoV-1, if you want to call it that. It utilizes the ACE2 receptors for cell entry and targets cells such as the alveolar epithelial type 2 cells, lymphocytes, but also extrapulmonary tissues that can be found in the heart, kidney, intestines, and other organs. In order to help us deal with the pandemic, it is important for physicians and patients alike to know who is infected. For that reason, the demand for COVID-19 testing has been rising over the past year. We all know that vaccines are on the horizon. Therapies have been developed, but vaccine deployment is taking time, and we will have to deal with COVID-19 for some time to come. There is a rising number of COVID-19 cases around the world as we prepare for a third wave of infections. Right now in the US, we can see a little bit of a drop and we're feeling a relief. However, we need to be ready for the third wave. Since we all have been locked up in our homes and have had to change the way that we are living our daily lives, some of us are starting to experience fatigue, both fatigue from just taking up the precautionary measures that are recommended by healthcare professionals, such as social distancing, masking, frequent hand washing, but also some of us are experiencing quarantine fatigue as we have been locked up in our homes for a long period of time. Nonetheless, now is not the time to let our guards down. It's important for us to double down and protect ourselves and our loved ones. And testing, therefore, is becoming more and more of a tool that we can utilize until we all get vaccinated. For that reason, Beckman Coulter, over the past year, has made it our mission to provide a menu that matters for COVID-19. As you can see on the schematic on your screen, as the infection runs its course during the early phases in stage 1, when only mild constitutional symptoms can be observed, such as dry cough, diarrhea, headache, etc., biomarkers such as D-dimer and LDH can be on the rise. Another biomarker and important indicator that we test for is the viral antigen for COVID or SARS-CoV-2 specifically. As we're progressing through the infection and the infection takes its course, stage 2 oftentimes is considered the pulmonary phase. During this phase, patients may be experiencing shortness of breath and chest images may look abnormal. During that time too, PCT, procalcitonin levels, may be abnormally low. During the third phase of infection, called the hyperinflammation phase, in severe patients, sometimes cardiac failure can be observed as well as ARDS or SIRS. During that very critical phase in seriously and severely ill patients, oftentimes IL-6 is observed to be an elevated biomarker. It's one of many inflammatory biomarkers that our body releases during hyperinflammation phase. Also, oftentimes troponin levels can be elevated during the hyperinflammation phase as well. Post-infection testing that often is done is related to antibodies such as IgG or IgM or both IgG and IgM together. Many of these biomarkers are being tested for by physicians in severely ill patients, but also in moderately ill patients. We can do some testing on antigen as well as antibody biomarkers as well. As you can see, it is critical that the laboratory is involved in testing as we're going through this pandemic. Point-of-care antigen tests are resource intensive. Oftentimes they're being utilized in order to determine whether or not a patient is acutely infected. However, high volume testing with point-of-care tests can be difficult to scale. Point-of-care tests are labor intensive, and the workflow can break down due to many steps involved in the testing process. This testing, point-of-care testing, oftentimes needs to be done by the healthcare provider themselves, which takes away from critical time that they would like to be spending with the patient to give them care and comfort. Point-of-care testing is wonderful and very important to utilize. However, it can be very difficult for the healthcare providers to scale it to deliver results for hundreds or thousands of individuals as they're coming through their doors. In some instances, point-of-care testing may require manual recording of patient data into electronic medical records or by some other manual processes, which again can take away time from patient care that the healthcare providers would like to spend in order to deliver treatment and comfort. For all those reasons, laboratory testing can alleviate some of the pain and pressure points on the healthcare system and support the healthcare providers during their critical mission of providing excellent patient care. For that reason, we want to offer reliable, affordable, and frequent testing to our laboratory partners that can assist them in giving confident results to the physicians and clinicians, to the nurses that are dealing with the patients themselves. As such, Beckman Coulter has developed a new SARS-CoV-2 antigen assay that strives to deliver highly accurate results comparable to most molecular tests such as RT-PCR. The Beckman Coulter SARS-CoV-2 antigen assay is currently under review by the FDA. Our performance data has shown that positive percent agreement for this new antigen assay at 7 days post-symptoms onset is 93%. Negative percent agreement is 100%. This is both true in symptomatic patients. However, we all know that asymptomatic patients require testing as well in order to understand whether or not they need to take quarantine measures to prevent further spread of the virus. For that reason, we have also performed performance testing with our assay in asymptomatic patients and have learned that the sensitivity level for access SARS-CoV-2 antigen in asymptomatic patients is 92%, and the negative percent or specificity is 100% in asymptomatic patients. This assay is primarily set to help you fight the pandemic by allowing you access to high volume testing. As we're experiencing shortage of PCR tests in some areas across the globe, antigen testing certainly can be a suitable alternative if performed frequently. Antigen tests can help laboratories conserve PCR tests for patients, but negative antigen test results are inconsistent with the clinical context of the patient presenting at the clinic or the hospital. They can be a wonderful frontline alternative because of great availability and low pricing. Also, antigen assays can deliver results in real time. They are able to provide you test results within minutes when sometimes RT-PCR tests, depending on what type of testing method is being applied, require multiple hours or even in certain instances a day or two to return results back to the clinician. It is important for us as a healthcare supporter to ensure performance of our assay has been tested to provide data to support performance claims. As such, the Access SARS-CoV-2 antigen assay has been verified and validated in in-house studies to be usable for both patients with symptoms who are consistent with COVID-19 or patients that are asymptomatic. Antigen tests can be utilized also for individuals in high-risk settings that may require repeat testing in order to provide relevant clinical information that may allow us to minimize transmission. In summary, antigen tests can be run on the same type of individuals as molecular testing. So both those suspected of having COVID-19 by their healthcare provider for various reasons, including them being exposed to others or working in high-risk environments. Please know that the FDA since December is also strongly encouraging the use of antigen assay testing in persons or people who are asymptomatic as well, and we have provided performance data in our instructions for use with that patient population as well. Secondly, it's important to have confidence in antigen test results. Confirmatory PCR testing following a negative antigen test in most cases is not necessary, especially not when the antigen test results are consistent with the patient presentation and if the pretest probability is low. Confirmatory PCR testing may be required if a positive antigen test is not is consistent with pretest probability, however. So you can see that really antigen assay testing can be utilized in the same circumstances as molecular testing. A benefit of antigen assay testing for the laboratory certainly is that it can improve your workflow efficiency by allowing you to run COVID-19 antigen assay testing alongside all of your other testing needs in true random access mode. With the SARS-CoV-2 antigen assay test, there is no need for batching or extra maintenance. You can truly run this assay alongside other biomarkers you may be checking for, such as the ones previously mentioned that are running their course through the infection period. After incubation with 0.5% Triton X100, which is part of the extraction agent that we are providing, no virus was detected in studies run at the British health authorities. Extracted samples, however, should be treated just like any other IA samples and should be considered infectious and handled accordingly. In order to showcase some of the differences between high throughput antigen assay testing versus point of care testing, I just want to point out to you in the audience how the test flow is working. The healthcare provider, denoted here on the left-hand side of your screen, will be collecting a swab just like in point of care testing. At that point in time, the healthcare provider will be placing the swab in a transport medium specified by the manufacturer. The transport medium will then transport the patient sample to the laboratory, at which point in time the laboratory is going to be taking off all subsequent steps needed in order to provide a test result to the clinician. The healthcare provider in the scenario of high throughput antigen testing can focus on patient care as well as the result report out subsequently and triaging of the patient in an appropriate manner. In comparison, if a point of care lateral flow antigen assay test is being used instead, the nurse or physician will have to run through not only the sample collection that you can see is equivalent in both scenarios, but will then also have to prepare the sample and run the assay as well as read out the test results and interpret the test results before reporting them out. You can see that there are multiple steps necessary in the case of lateral flow point of care testing that involves the healthcare professional taking away potentially time from handling the patient's concerns. In summary, high throughput antigen assay testing can provide accurate results that are supported with performance data in both symptomatic and asymptomatic populations. In the case of the Beckman Coulter SARS-CoV-2 antigen assay, we can detect lower quantities of virus than some of the currently available point-of-care tests, with a limit of detection in our case of 33. High throughput antigen assay testing is fast and allows for a large throughput per hour. In the case of Beckman Coulter, you can run 50 to 200 tests per hour. We feel that we can allow and help you to optimize the workflow for mass screenings. Healthcare professionals only need to collect patient samples and do not need to process the samples afterwards. All of those steps are moved to the laboratory that is set up for high throughput testing. On the other hand, if you decide to utilize point of care antigen assay testing, which also delivers high performance and accurate results, you may have to take on the flip side the case that only low throughput is available to you. In many cases, point of care tests only allow you to run about 5 to 20 tests per hour, as they are not optimized for mass screening. The healthcare professional not only needs to collect patient samples, but in most instances also process the samples and interpret the results as well as report them out. Antigen testing can be utilized for different purposes. The reference for the slide that I am presenting to you now is the CDC. It highlights the use cases for antigen assay testing, both in order to diagnose symptomatic patients as well as to be proactive and screen asymptomatic patients on the right-hand side of the screen. Antigen testing is certainly appropriate for symptomatic patients when access to PCR or molecular testing is limited. Antigen testing can be utilized to be proactive in screening asymptomatic patients in the case of healthcare professionals, in research centers, and for epidemiological studies, and can also be applied by governments and NGOs to expand testing and support better decision-making as it relates to reopening our economies across the globe. In summary, I want to close out the section on antigen assay testing by giving you an overview of the Beckman Coulter Access SARS-CoV-2 antigen test. As it relates to throughput, you have, depending on your analyzer, the capability to run between 15 to 200 tests per hour. The time to first result for this antigen assay test is rather swift. It takes about 30 minutes. Patient samples do not need necessarily to be refrigerated. If you're sampling and testing the patient sample within a period of 48 hours, the patient sample is stable for in the refrigerator. The patient sample is also stable at room temperature for up to 24 hours. During our performance validation, we have understood that sensitivity for our Beckman Coulter Access SARS-CoV-2 antigen assay in symptomatic patients is 93% and in asymptomatic patients is 92%, with a specificity profile of 100%. Results of the Access SARS-CoV-2 antigen assay are comparable to RT-PCR assays in 7 days post-symptom onset for symptomatic patients. Now let's move over and talk about another assay in the COVID-19 toolbox that we would like to present to you this morning. It is called the Access SARS-CoV-2 Immunoglobulin G2 assay. And Alex, I believe you may have a poll question for the audience. The Access SARS-CoV-2 antigen assay is a semi-quantitative product that measures a patient's level of antibodies in response to a previous SARS-CoV-2 infection. We know that it's critical to understand which patients are carrying antibodies to the disease. Our assay, the SARS-CoV-2 IgG2 assay, provides a numerical results in arbitrary unit in the range of 2 to 450 arbitrary units per milliliter, as well as a qualitative result giving you an indication whether a patient may have previously been exposed to the SARS-CoV-2 virus and subsequently developed a convalescent response and antibodies to the virus. The Access SARS-CoV-2 immunoglobulin G2 assay is a semi-quantitative antibody test that allows clinicians to establish a baseline for antibodies based on a numerical result. This allows them to assess relative changes over time of an individual's immune response to the SARS-CoV-2 virus. Studies determining the rise and fall of antibodies to the SARS-CoV-2 virus may be relevant in order to develop therapies and also understand vaccination response. The Access SARS-CoV-2 IgG assay can be run on all Beckman Coulter Access instruments. It allows you to process up to 50 tests per hour on the Access 2 analyzer. If you're choosing to utilize the DXi 800, which is a larger instrument, you can run up to 200 antibody tests per hour with the Access SARS-CoV-2 IgG2 test. The readouts that you will be getting are both numerical as well as qualitative. You will see that results coming back greater or equal to 10 arbitrary units per 10 milliliters are going to be reported as reactive to the SARS-CoV-2 virus, and we will be able to deliver a level of antibodies in response to those SARS-CoV-2 viruses in the patient samples. Any test results that are coming back with a reading below 10 arbitrary units per milliliter are going to be considered non-reactive to the IgG antibodies. The assay has been tested rigorously, and we have found that 15 to 60 days post-symptom onset, Access SARS-CoV-2 IgG2 has a sensitivity profile of 98.9%. Specificity as validated in 1,448 patient samples has been determined to be at 99.9%. Our Access SARS-CoV-2 IgG2 assay targets the spike protein that initiates our bodies to make antibodies and generate the T-cell response against the SARS-CoV-2 virus. It assesses relative changes of IgG levels over time. This helps you to understand the durability of an immune response. It's imperative to utilize high-quality laboratory tests in the battle against COVID-19. The CDC recommends 3 stages to improve positive predictive value and ensure that you are dealing with a high-quality assay. First, the CDC recommends you choose a test with very high specificity, perhaps 99.5% or higher, and a greater sensitivity profile in populations with prevalence that may be laying above 5%. Secondly, persons with a history of COVID-19 like illness who have or may have a high pretest probability of SARS-CoV-2 may have developed antibodies. Third strategy to improve positive predictive value in antibody testing is to employ orthogonal testing algorithms in which persons who test positive are tested with a second test to just ensure that you're giving the right readout to your patients. As I mentioned earlier, the Access SARS-CoV-2 antibody test, here in this case IgG, but all of our antibody tests are targeting the S protein. The S protein has been identified as an ideal target not only for antibody testing but also for vaccine development. Neutralizing antibodies are important to understand their therapeutic use of immune plasma and vaccines. In SARS-CoV-2 1 as well as SARS-CoV-2, vaccine proteins are interacting with the ACE2 receptors, and antibodies targeting the spike protein are interfering with the binding. For that reason, they have been found in laboratory studies to be neutralizing to the virus and binding the blocking between the virus and the ACE2 receptors. To monitor immune response post-vaccines to the SARS-CoV-2 virus, the antigen target of tests is extremely important. We pride ourselves in targeting the RBD of the spike 1 protein. On this slide, you can see that, for example, other vaccine manufacturers are also targeting the S1 protein. For example, Pfizer and BioNTech's vaccine is causing our bodies to produce a viral spike protein. This spike protein initiates our bodies to make antibodies and generate the T cell response critical to protect us against the virus in the future. Researchers are using tests that target the same antigen as the vaccine to understand level of protective antibodies produced. Researchers also are using tests that are targeting the same antigen target as vaccines to assess the change of levels over time and demonstrate the durability of an immune response to the SARS-CoV-2 virus long term. I believe on the previous slide we had a poll, so I'm going to open it up for that and move on to the very last slide of my presentation to summarize this morning that we are striving to provide you with reliable, affordable, and frequent testing options in order to fight COVID-19. We hope that together we can take care of our community's health with antigen testing, as well as IgG and IgM testing tools to identify individuals who are actively infected or have had a previous infection in the past, so that we can continue to understand the disease of COVID-19 better and curb the pandemic to return back to a more normal life very soon in 2021 and beyond. Thank you for your attention this morning. And with that, I'm going to open it up for questions and also give you a very final poll question. If you don't mind answering it, that would be great. Thank you very much.
Thank you so much, Jellica. So with that in mind, we're just going to do a few of the polls actually. If you can just answer them, and then we'll have a little bit of a discussion. So here's the first one. So the question was, how likely are you to use the throughput antigen test? Most of you, or the majority, said very likely. So this is the first response to the poll. Jellica, do you have any comment on this before we move forward?
Yes, Alex, thank you. I'm very happy to hear that there is a high interest in utilizing high throughput antigen tests. I understand that with regards to high throughput antigen testing, we are really opening up the opportunity to offer much more testing to our communities and take the appropriate measures. So thank you very much for that feedback. We're glad to hear that this is meeting the mark.
Great, thank you. So with that, we can go on to the next poll. Angelica, just for you to know, this one is, will you use antibody testing for post-vaccine follow-up? We'll just wait for the answers and we can have a little bit of a discussion.
Thank you. Over the course of the past few weeks and months, we have often heard the question and the request around post-vaccine testing, and it will be interesting to see how many of our audience are intending to do post-vaccine follow-up. At this point in time, there are few studies available on antibody testing after vaccination, really both pre and post-vaccination. So we're eagerly awaiting to see studies be published and learn more about how our antibody levels may be changing after vaccines.
Very interesting. Thank you. Just for you to know, actually, I would say 91% said yes to the question. So basically, yes to understand the durability of the vaccine. So that was 91%.
Thank
you.
You
Go to the next poll now. So the next poll is, what type of antibody test are you most likely to use? And of course, some of the answers are total antibody test to check for IgG and IgM, quantitative IgG antibody test looking for spike proteins, IgG antibody test looking for nucleocapsid. Sorry if I said that. I hope I pronounced that correct, and other. So we'll just wait for the results to flow in.
Yeah, this is going to be an interesting poll. It will be interesting to learn more about what type of testing is going to be the most suitable post-vaccine. At this point in time, throughout the pandemic, we have started learning more about what happens to us after being exposed to the SARS-CoV-2 virus. And there have been different studies outlining that in some patients, the IgM levels are rising pretty early, which would be consistent with other serology studies and knowledge that we've had in the past. However, in some individuals, IgM never really rose, and they developed IgG antibodies right off the bat. In some individuals, we have seen IgG rising first, then IgM. So it will be interesting to see what the audience would like to do as far as post-vaccine follow-up is concerned, and understand the state of their patients.
Thank you, Jellica. Just for you to know that the results of this question were 23% voted total antibody test to check for IgG and IgM, and 73% voted for quantitative IgG antibody test looking for spike protein, and 4% voted IgG antibody test looking for nucleocapsid.
Interesting. Thank you very much. That kind of jives with the slide that I shared earlier, where the vaccine manufacturers are targeting the spike protein. For that reason, it sounds like our audience, of course, would want to know whether or not they have a antibody that is, or a test that is looking for an antibody targeting the spike protein as well. Really interesting. Thank you.
Thank you. And I believe we have just one more poll.
Mm-hmm.
All right. So the last
I believe
one,
it's around what type of vaccines are being used. Is that possible?
exactly. So what type of test? Well, no, actually, what type of testing do you do? So it's point of care antigen testing, IgG antibody testing, PCR testing, and laboratory antigen testing.
Mm-hmm. Yeah, I suspect that our audience is probably utilizing all of the testing options, but it will be interesting how it breaks down.
All right, so the results are in, and 10% voted for point of care antigen testing, 20% voted for IgG antibody testing, and 80% voted for PCR testing. Now, 7% for laboratory antigen testing. Of course, this was select all that you apply, so that's why the percentages don't add up to 100.
Great. Thank you very much, Alex. Thank you for letting me know. Not a huge surprise there as it relates to PCR testing. That definitely makes sense. It's the gold standard. We know that in the past, PCR testing may have had some supply issues just because we were trying to ramp up as it relates to PCR testing. Now that it's more available to the masses, no surprise there. I hope that that the supply continues to be available and that our partners continue to fight the pandemic utilizing the right tools and taking advantage of the mix that is available to them, including high throughput and POC testing as well. Really interesting polls. Thank you.
Perfect. Now we're going to come back to the poll that we shared throughout the webinar, which was the one that you just mentioned, Angelica, which is which vaccines are currently used in your institution. So we already have the results here. And the results are 71% have stated that it's Pfizer or BioNTech. 10% is Moderna. And 35% is AstraZeneca, and we have 29% that's other.
That is a large amount for other. Very interesting poll. Thank you very much, Alex. I wonder whether this other is related to the Russian or the Chinese vaccines, but we didn't have the opportunity of having other actually be filled out. Really interesting. And again, I am not really surprised to see the breakdown of the other three large ones. It seems like Pfizer, since it was the first one to market in most of the markets across the globe, is really gaining traction. Thank you very much for doing those polls and providing the results to the audience. I hope it was interesting for everyone who dialed in.
Thank you. I'm sure it was. Now, with that in mind, we'll just go to the question and answer part of the session.
Sure.
If we don't get to your question, we will be emailing you the results later on. So please bear with us. So the first of the question that came in, is it possible for this test to give a negative result that is incorrect? For example, a false negative?
Yeah, in general, positives are highly accurate. And there is a range of false negatives depending on when the testing is performed during the course of an infection. So no test currently available that we're providing, is 100% foolproof. You can see that none of the tests that I presented to you had like 100% sensitivity and specificity. So false positives in COVID testing, just like in any other laboratory testing, can occur, but they are extremely rare when you have a positive test result, and a negative test result can sometimes be negative depending again on when the test is performed during the course of infection. Great question. Thank you.
Great, thank you. The next question is, what is the recommended type of transport media that frozen samples should have been stored in for validation purpose? So for example,
our assay.
Sorry, go ahead. No, please.
So our assay is an assay that has been validated and verified in testing with 5 specific brands that should be utilized. in accordance to the instructions for use. To name a couple, Remel, Copan, and Bartel are some of the ones that we have validated. It is, however, possible, if you don't have access to those transport media, to validate and verify an alternative solution that is available in your country to utilize with the antigen test. The transport media are only specific to antigen assay testing. You mentioned in your question also whether the sample should be transported in a frozen state. That is not necessary. You can keep the sample at room temperature for up to 24 hours, in the refrigerator for up to 48 hours, and then we have an extended time of up to 30 days in the frozen state. I suspect that because this is COVID-19 testing, most of the samples will be tested immediately, so you wouldn't even get into the frozen state at all. Again, 5 different transport media have been validated and should be utilized according to the instructions for use with the Beckman Coulter Access SARS-CoV-2 antigen assay.
Thank you. Thanks a lot.
Mhm.
So the next question is, does sensitivity of antigen test decrease the time after symptom onset?
So if you're looking at serology in general, typically what happens is that a viral antigen starts to rise after the early infection periods takes place. So after a couple of days, you can see the viral antigen increasing. At some point in time, our body kicks in and we're starting to develop antibodies such as IgM or IgG to fight the virus. And at a certain period of time, for that reason, the viral antigen is going to decrease. Depending on literature and performance testing, you can see that the levels of sensitivity and specificity, depending on the course of infection with an antigen assay, can vary from day to day. And they're slightly increasing as you're getting, as you're actually starting to produce more of the viral antigen before ebbing down again. So the testing will depend a little bit on the evolution of the infectious process.
Perfect, thank you. So we have 3 more questions. The next one is, what is the difference between policy C and policy D?
Okay, so we're looking at the regulatory process in the United States. So in the United States, because we are in an emergency situation right now, we're able to get COVID-19 related tests to market without getting a 510K that you would typically be seeing in the IVD industry. The alternative path is to receive what is called an emergency use authorization. Prior to receiving emergency use authorization, in order to provide the products necessary in order to fight the pandemic, there are 2 policies available to manufacturers that have a lot of guidelines associated to them that allow you to market your product prior to receiving the emergency use authorization. Policy C is the pathway that manufacturers take in order to get a diagnostic test to market, such as the antigen assay that we spoke about. Policy D is reserved for serology testing, and the requirements under policy C and policy D are different because you're looking at different types of tests.
Thank you. And so the second to last question we have for this evening, or your morning, Jellica, also I'd like to thank you for waking up super early and helping us with this.
My pleasure.
So the next question is, how would you imagine using this high throughput antigen testing in airports and port of entries? This is very interesting.
Yeah, that's a great question. So as you may remember from the workflow slide that I shared with you, there is only the need to swab a patient and then transport that swab to the laboratory to then quickly run a large number of tests on an analyzer, up to 200 tests with the DXI 800. So what is potentially feasible to do as we're looking at airports or port of entry is swabbing could be performed while the passengers are still on the airplane, put into the transport medium, and then the samples could be very quickly taken to a laboratory on site at the airport potentially, and the tests could be run there in high volume so that you would get up to 200 test results within an hour back and know what the infectious status is of the whole airplane that you're testing, as opposed to having people standing in line and waiting to go through the process and having each individual sample, in the case of a point of care test, take about 15 to 30 minutes from time the sample is received to being processed. So perhaps a turnaround where you can just like have a larger volume of swabs being taken in a more convenient setting and then transferred to a laboratory that then runs the tests and would push back the results to the individuals electronically. It's just, an idea. I don't know that it's necessarily feasible. You're just asking me hypothetically, what could a scenario look like? So that is one way of potentially thinking about it.
Perfect. Thank you so much. So actually, we have two more questions. Another one came in on the question and answer or the question section
on
of
the chat.
webinar. Yeah, exactly. So when will IgG2 be quantitative?
Yeah, as soon as the performance testing has been run and we submit the technical file to the governing bodies, you'll be able to utilize the IgG2 assay in a quantitative manner. The WHO just launched or just provided the standards for the first international standardization. So everybody is looking to just do the conversions and the testing required in order to have a standardized result report out as compared to the semi-quantitative result report out that was possible prior to having a worldwide reference standard with a reference material that could be used across different manufacturers. So this just recently came out in December.
You must be a mind reader, because that was the second part of the question. Which was, do we have some standardization for the test according to who? And of course, with the units of BAU or ML. That was the last part.
Can you repeat the last part of the question?
With units in BAU or /ML?
Okay, so the units right now for our IgG2 assay are arbitrary units per milliliter because we set a standard internally for our assay. So that's why you saw the AU over ML in the slide deck. Because the World Health Organization has set a standard, the units going forward, once there is a fully quantitative assay reference back to the WHO guidelines, will no longer be arbitrary units. They should be conforming so that then you can compare test results between manufacturers. Right now, you can only compare the test results for one individual, in the timeline with the usage of one specific manufacturer. You can't go between tests, but once tests have been brought to market that are in accordance to the WHO standards, they are standardized, and then you can make comparisons between individuals and between tests. And it will be concentrations per milliliter, but not necessarily the arbitrary units. Arbitrary unit is just set by the manufacturer.
Okay, perfect. And the last question we have today, and then I think we're just about done with time, is will I get a positive antibody result soon after my vaccination?
We have not performed post-vaccine studies with our assay as of yet. Our antibody assays, both the IgM as well as the IgG assays, are targeting the spike protein. And as you saw, vaccine manufacturers are also using that same antigen target. However, we don't have published data on post-vaccine follow-up at this point in time. We hope that that will be coming very soon now that vaccines are starting to roll out.
Great, thank you. And that was the last question that we have. So
Great, thank you very much.
Thank you so much. With that, I'd like to thank you, Jellica, for your time again today. It was a great session, and we look forward to having you again speak in our region. And I'd like to thank the audience for giving us an hour of their time. So thank you very much. And with this, I'd like to close the webinar for today, and everyone have a great rest of day or evening ahead of you.
Thank you very much. It was my pleasure. Stay safe, everybody.
Stay safe.